So our first speaker is going to be Morten Lietz, and he's a PhD student at the University of Fribourg. Also a colleague as a member of ALPS, where he's coordinating the speakers for the conference. And by the way, yeah, a lot of the speakers today seem to be associated with ALPS in one way or another. I just want to mention here at this point that ALPS did not choose or vote for the speakers, so it's not, the speakers aren't favored towards ALPS members. If anything, I mean, the Swiss Psychedelic Student Network, with representatives from all the student associations, voted for the speakers in the end. And if anything, the connection would be, in my opinion, that those people that are particularly intrinsically motivated are just not only more likely to be already researching psychedelics during their studies, but also more likely to join ALPS. So that will explain it. So back to Morten Lietz. Today, he's going to talk about neuroplasticity, which is also the main focus of the research lab in Fribourg. And neuroplasticity is currently a big topic in psychedelic research, and Morten is going to shed some light on it and hopefully help us understand what it's all about. So please join me in warmly welcoming Morten Lietz on stage.
All right. Always great to start on a note of that the reason I'm here is not nepotism. Amazing. Yeah, thank you, Dave, for the warm welcome. I'm going to tell you a little bit about psychedelics and neuroplasticity. First, to get you all on the same page, what do we mean when we talk about neuroplasticity? Well, the formation of new and the alteration of persisting neuronal connections. This is one way of looking at it. There are different definitions out there, but for the sake of simplicity, let us stick with this one. So let's look a little bit more at how plasticity works. Again, this is a more broader overview I'm going to give you here because in the nitty-gritty stuff it's a bit more complex. But like the basics are these three rules. They're called Hebb's law. Hebb was the guy who discovered these. So we start with simultaneous activation. So synapses that fire together, wire together. Temporal associations, so the timing there matters as well. They don't have to fire exactly at the same time. It's already enough if they fire in close proximity, because that's one way for your brain to interpret that this is a meaningful connection, so it's worth to invest resources to actually keep this connection. And it's use dependent. So basically, imagine your neurons like a muscle. If you use them, they grow stronger. If you don't use them, they atrophy. Same principle holds on here as well. Now, let's look a bit about the role of neuroplasticity in a broader context. Well, neuronal plasticity is almost involved everywhere because it's basically how your brain reacts to a changing environment. Now, as you can see, there are, we can't really see this, but that's okay. So some studies have pointed out that there are neuroplastic changes in neurodegenerative diseases such as Alzheimer's, Parkinson's, MS. Has been associated with psychiatric disorders such as depression, but also drug abuse, schizophrenia, neurological disorders such as traumatic brain injuries, stroke, chronic pain, but also in neurodevelopmental phenomena such as OCD or autism. Interestingly here is that it's not only going in one direction. So when you are on this page somewhere here, it doesn't mean that your neuroplasticity is reduced. Sometimes it can even be higher than normal in specific areas of your brain. So neuronal plasticity does not mean good or bad. It only means modulation. So basically the opportunity for change. And that can be a good change, that can be a bad change, that can be completely meaningless change. So let's see what psychedelics do in that field. Well, quite a lot, actually. So there is this idea that psychedelics are kind of transdiagnostic in some way or another. So, for example, when we look at the roots of this tree, this one doesn't really work. Well, if we look at the roots of the diagram I made over there, which kind of represents the more fundamental research that has been conducted with a more rigorous design, so this is good stuff, this is clinical trial design, we already identified several different disorders or domains in which psychedelics seem to be helpful. For example, PTSD, addiction, depression, cluster headaches, and end-of-life anxiety. And in the leaves of the tree, you have the more ongoing investigations. The reason why I made this distinction is because these are basically just like very early on studies. We don't really know whether or not this will hold true if we conduct this research in a bit more rigorous manner. So these are mostly open label, small scale. So take all of this with a grain of salt. However, when we look at this and we compare that to the previous diagram, we can already tell that there's a bit of an overlap there. And some researchers have suggested that the main reason why psychedelics are so transdiagnostically helpful, which means that they're applicable for so many different disorders, is because they seemingly tackle like an underlying issue that is the same within all these disorders. And questioning the methods of what that could be would be, for example, neuronal plasticity. And that's kind of why I'm talking about this today. And let's look a bit at the mechanisms. So neuronal plasticity can be measured at so many different levels. Let's start off with the more lower levels. For example, on the molecular level, we can just simply look at genes that have been associated with neuroplastic change. In order to do so, we can just run a PCR to see whether or not these genes are there in a sample. We could also do something called northern blotting in very probably wrong terms. That means you take your cells, you put them in a blender, and then you simply put them on like a sheet, and it tells you how much of a certain gene is in that piece of sample. We can also look at different protein abundances at the synapse level. One way to do that is using a PET scan nowadays. That's quite a cool new development that is coming out. We can radio label certain proteins within your brain, and then the scanner tells you exactly how much of that protein is in a specific region, which is kind of wild, considering that this is a PET scan. So you can do this in an alive human being, which is pretty awesome. But also the more classic methods of looking at these things is through a microscope. And of course, what we also can do is like voltage and cell patch clamping, which is a pain in the ass to do, I have been told. I have never had the pleasure of actually doing this. But you stick electrodes in different neurons and you measure how they are communicating with each other. And this change in communication would be like a level, would be a measure of neuronal plasticity. But what I personally find most interesting, because I'm a neuroscientist who does not spend so much time in the lab with the lab coat on, but rather with people, is of course the more network, brain, and behavioral level of neuronal plasticity. So there are several different things we can look at here. We can look at network connectivity, for example, so how different parts of your brain communicate with other parts. We can look at the brain as a whole. We can do an MRI scan and basically compare density of tissues in specific regions. Or we can look at behavioral outcomes. So learning and memory, for example, but also cognitive flexibility, behavioral flexibility, and so on and so forth. So this is just to give you an overview of how many different things you can do to look at something that may or may not be related to neuronal plasticity. The answer is virtually everything. Well, not everything, but a lot. It's a lot. So how does it work on a cellular level? Well, probably not like this. This is a very overly simplified way of looking at this. However, this is still the easiest you can do for an audience that is not really on a molecular scale yet, which is why I choose this picture again and again. So remember from before, what wires together fires together. Similarly, when you have co-activation at a synapse, then this will probably induce some form of neuroplastic change. The way this is working with psychedelics is psychedelics are, as you probably heard by now, 5-H2A receptor agonists. That mean they attach to the receptor, which is the 5-H2A, which is a serotonin receptor, and they activate that receptor. Now, what happens in this particular model is that you have these deep cortical layers where this neuron gets triggered through this receptor, which then leads to like an action potential that travels down to its synapse, and there you have this efflux of glutamate, which is also stimulating the synapse. Now, at the same time, you also have these serotonin receptors roughly at the same cell, so you have this strong co-activation. Now, like a strong activation at a cell, specifically at a neuron, usually means for the neuron to be like, okay, so this is like a beneficial change that is happening. Let's do something. We want more of this stuff. And it leads to like a signaling pathway that ultimately increases, for example, gene subscription or like the production of a certain protein. So how does it look over time? Well, first of all, this is the results of my colleague Abigail Calder. She has been conducting a very thorough meta-analysis, systematic review on neuronal plasticity and what has been published so far, and she has identified that actually the neuroplastic change can be measured over quite a long period of time. In this particular instance, for example, up to 30 days after the administration of a psychedelic drug. So there seems to be the case that you give a psychedelic which opens like this neuroplastic window. This is also something that has been found by Nardu and colleagues. They were looking exactly how long this window of neuroplastic change remains open, and what they found was quite interesting because seemingly the duration of the psychedelic effect in humans kind of more or less maps on what they found in these behavioral, more neuroplastic stages in mice. In terms of how long these windows of opportunity were open, how long this neuroplastic effect lasted. So when you look at ketamine, for example, which is like 30 minutes to 2 hours, the window remains open for like 48 hours in mice. However, when you look at LSD or ibogaine, it's more like 3 weeks. Ibogaine was even so long they didn't find like a decline during their measurement period. Take all of this with a healthy grain of salt because these behavioral paradigms in mice, who knows how that translates to humans. However, this was already quite interesting, and this is an interesting model to work on specifically when we look at clinical applications later down the line. Right, so now I'm going to zoom into the different levels of how to look at neuronal plasticity. Let's start at the molecular one. I promise I'll keep it light. So first we look at psychedelics and culture. Of course, this is not what I mean with psychedelics and culture, although this is a very cultured mushroom. Of course, I'm talking about psychedelics and cell culture. Unfortunately, the layout is slightly different here than it was before, but, well, doesn't matter. So what I mean with cell culture, for those who don't know, well, you basically scrape a bunch of cells of interest into like a bowl with a certain form of liquid that these cells need to grow and multiply. Maybe you put in some hormones so that these cells make more of themselves. Then you stuff them in like an incubator, which is like a reverse fridge, so it keeps your stuff warm rather than cooling it down. And over time, you will have like a very nice amount of cells of interest within your cell culture. You put them onto these plates. And how do you look at what psychedelics are doing in that thing? Well, you basically just add psychedelics and see what happens. And this is exactly what the research group was doing. Oh, man. So Lai and colleagues, you already saw this in the previous presentation, but they were doing exactly that. So they had cell culture, they applied some psychedelics, and they used a super expensive microscope, which is a confocal laser microscope, costs like $1 million, it's amazing, to make these super pretty images. And well, very simply put, when you look at on the left side, on the very top image, vehicle means salt water, basically. Salt water doesn't really do anything to the neuron, however, DOI, which is also psychedelic, but also DMT and LSD, which is probably more known to you guys, seems to induce some form of change. So the neurons become more complex, in simple terms. And yeah, basically to sum it up, they made this very nice graphical abstract. So basically, psychedelics do increase dendritic arbor complexity, which is like a measure of these things. So how complex the whole, well, the branching of the tree, so to say, looks like. It promotes dendritic spine growth, so how many potential areas where another synapse can dock onto are there, and they stimulate synapse formation just straight up. So that was already quite interesting. Right, so keep in mind this was also on cells, not from humans, but from Drosophila, so fruit flies and mice. So let's take a bit of a jump and talk about the network level, which is something we're currently also working with in the lab in Fribourg. More precisely, we mostly work with EEG. And how do you use EEG to assess neuroplasticity in humans. This is what I'm going to talk about now. So, well, in simple terms, what you do is first you take a baseline measurement. So no psychedelics, no stimulation. You simply take a baseline measurement in which you give a certain stimulus to a participant. Like this could be either like a checkerboard that flickers at a specific frequency, but this could also be a tone, and you measure the brain's response. Now, in the next part, you'll try to induce an effect within the brain that upregulates its sensitivity towards a certain thing. In other terms, you try to induce something called LTP, which stands for long-term potentiation. So you try to sensitize basically this part of your brain that is responsible for the certain stimulus. So what happens now is you use this either like a tone or you use like the flicker board and you just blast the participant with it for 12 minutes, 15 minutes, depending on the paradigm you're using, but you just basically blast the person for quite some time. And the brain gets kind of used to it and it realizes, okay, so we keep getting the same activation in this area, so maybe this is important, so let's upregulate our responsiveness towards that specific stimulus. And this is what you do in the middle part, and then afterwards you basically do the same thing as before, you measure again, and what you hope to see is something like this. This is from the paper of the guy who developed this in humans, and what you can see is that when you compare the baseline, which are these levels, these curves over here, with like the post-stimulation, you have this nice little increase, which means that there was something that looked like an LTP happening within that region. So your brain becomes more responsive to that stimulus. Now, very simply put, and how do we use this to measure neuroplastic changes after LSD? Well, part of your sample gets LSD, the other part doesn't. You compare, and you kind of hope to see an increase in both of them. However, you hope that the increase in the group that got psychedelics is larger. And that's how we index neuroplasticity in our study. We're not the only ones who are currently doing this. This already has been done. And man, that's kind of sad. So this already has been done using psilocybin and major depression disorder by this group by Skonsnik, I think it's called. So they used 19 patients who suffered from depression, who are either currently under moderate or severe depressive episodes. These guys had to temper off medication because, well, SSRIs also have an effect on neuroplasticity, so you don't want to mix that up. They got a medium dose of psilocybin, which was roughly like 20 milligram for not for me, I would have gotten more because I'm a bit heavier than 70 kilos, but you get the idea. And they used the same paradigm with auditory stimulation. These guys, they had like first a waiting period in which they got all the measures but only a placebo, and then after some time they repeated the same thing but this time with psilocybin. Keep in mind they always got therapy with this, so this isn't simply psychedelics, this is psychedelics in a therapeutic context. And what they found is that nothing really happened in these EEG measures after 24 hours. However, after 2 weeks, which is on the bottom diagram, the people who got LSD, which you see in red, have like a higher theta power level than the people who didn't. Well, basically, these are the same people, but like before they didn't get it, and now they got after they got psychedelics, right? And interestingly, this is also correlated with a decline in depression symptoms. However, there's a lot, I'm not going to say wrong, there's a lot of stuff peculiar with this particular paper. My colleague pointed out to me like a while ago when I first came across it because I was like, wow, this is cool results, this is amazing. So it's really hard to notice what is wrong with this paper if you're not really deep into the thing itself. First of all, theta power. Why theta power? Why? What does this have to do with neuroplasticity in that specific region with depression at all? They didn't really answer, and there's no apparent reason to use this matrix over the more commonly used ERPs, which is just like you look at a specific region and you basically just see how the amplitude is changing over time. You don't have to do all the frequency stuff. So the thing is, being an EEG guy myself, I know how many different analyses you can run on your EEG data, basically limitless. And the longer you run different analyses, the more likely you are to find something. I'm not saying that this is what they were doing. However, I was a bit, Abigail pointed that out to me to be a bit more skeptical about these things, which I very much appreciate. So, yeah. I mean, also, what could also be the case, if this is all nice and fine, dandy, maybe they just got better in terms of depression, and therefore this might also have been an effect on neuroplasticity. We don't know if this is directly related to the psilocybin. Anyways, let's look at the behavioral level. Now, we have seen that cognitive flexibility also seems to be a very nice matrix of neuroplastic change, because ultimately what we hope to see, of course, is that the stuff that goes on in your brain is reflected in how you act in one way or another. And, well, like a lot of people suggest that cognitive flexibility might be a good candidate for that matter. Cognitive flexibility basically means your ability to shift between different sets of rules, to adapt to like your environment, which seems almost exactly to what your brain is doing in its own soup, right? So they were looking into this effect, so Doss et al., they were looking into the effects of psilocybin again, on cognitive flexibility. Again, this is a depression sample. They had 24 patients. They got 2 sessions of psychedelic-assisted psychotherapy, once with a medium dose, once with a high dose. And then they did this Penn conditional exclusion test, which basically you have to follow different sets of rules and you point at different objects that look kind of out of line. And they find that, here you can see that in the graph on the bottom. So baseline is, of course, before they got therapy plus psychedelics, and then they measured this again 1 week after and 4 weeks after, and the error rate was reduced. So they got better in this task, which means they got better at cognitive flexibility. Now, interestingly enough, this, even though it looks like this should correlate a lot with a HAM-D score, which is a matrix of how strong the depression symptoms were, these did not correlate, actually. So seems to be that this effect is more or less independent from their lack in depressive symptoms, which might be suggesting that this is a direct effect of psilocybin. Well, of psilocybin plus therapy. Keep that in mind. Right. So what can we do with this? How can we utilize psychedelics' effect on neuroplasticity, if they are true or not, for clinical practice? Well, the one way to look at it is through this analogy. So let's think about your brain as being a pot in which you cooked rice and you kind of forgot about it and now it's all sticky. It's really annoying to get all the rice off. Well, you could use some form of intervention, for example, therapy or like some form of cognitive training or whatever to get all the rice off. But your brain resists to the change. Your pot does not want to be changed. This is exactly what happens in depression, for example. You kind of resist, you're kind of stuck in your mindset. You can't really change that easy, and that's why therapy is so hard and takes such a long time. Now, what you could do is you can use something that just loosens this up. For example, soap and water, and let it sit overnight. So you can pre-treat your dirty pot with something that makes it easier to implement the change you want to implement using a specific intervention. And along these lines, we kind of came up in the Netherlands before I came here with this project, which is psilocybin-assisted neurofeedback, PANF in short. So the QR code on the left will bring you to the preprint. This got accepted like a couple of weeks ago, which is my first author publication, which is awesome. Thank you. Thank you. Right, and in this case, the way we thought about it was basically, hey, let's use something that increases neuronal plasticity to begin with, and then follow up with an intervention, and maybe this can improve the effectiveness of this intervention. For this matter, we used psychedelic-containing, psilocybin-containing magic truffles, because they're legal in the Netherlands, and you can simply buy them. And our intervention was neurofeedback to train cognitive functions. So let's first start off with neurofeedback. What is neurofeedback? What do I mean with neurofeedback? Well, neurofeedback is quite an interesting piece of technology where you basically get like a readout of what's going on in your brain. This could be either through EEG or fMRI. And you get like a representational image of what is going on in your brain on screen in front of you. So basically you see what's going on in your brain. Now, how do you use this to train anything? Well, There are specific frequencies that are responsible for specific tasks. For example, in our case, we used FM theta, which is theta frequencies right here at the tip of your head. They seem to correlate with cognitive functioning. So the higher your FM theta, the better you are at specific tasks, because this is like the underlying mechanism. When your neurons are well in tuned and work very well, then you should theoretically do better in task, and this is what we've seen in studies. So this then gets read out and fed back to you, and you basically see, am I currently focusing on upregulating my theta or not? So how do you upregulate your theta? Well, you can do, for example, meditation exercise, but you can also do arithmetic, so try to calculate stuff. You can try image rotating within your brain, and you see on the screen whether this is regulating your theta or not. And that's the cool part, because technically what you're doing is you're training an underlying circuitry within your brain. Now, the idea of why combining that with psychedelics was basically, well, in order to get any change happen within your brain needs to undergo a certain form of neuroplastic change. And can we use psychedelics to increase neuroplasticity and therefore improve the efficacy of our modulator, which is the neurofeedback? So this was our study design. Now, before we go into this, big disclaimer up front. This is a feasibility study. We are the very first to attempt anything like this. Also, we were completely unfunded. We solely relied on volunteer work, and we had zero money for this, and basically no time, because we all had full-time jobs next to this. And still, we kind of just decided, you know what, fuck it, let's do this. And somehow it worked out. Well, first and foremost, because we got people from the Microdosing Institute on board. They were very helpful in providing the substance for free to the participants when they decided to work with us. And that's the nice thing, it's legal in the Netherlands, so therefore they could just get like a code from these guys, and then they could just basically through them source the substance, which are these ready-to-take microdosing truffles. They come in a 1 gram microdose bag, and you can just cut them open and take them. Now, we had 18 people in the experimental group, 19 people in the passive control group. These guys, they were also microdosing at one point, but we told them basically, we have to do 2 measures first, and then like another 2 weeks later, after you finish microdosing, we do a 3rd measure. So they still had the same amount of expectancy towards microdosing. They were also from the same population, so they were really keen on microdosing. They were really pro psychedelics, yet they didn't get the neurofeedback. That's like the main difference. So our experimental group, though, however, they first started microdosing for 3 times in order to find the right dose for them, followed up by continuing microdosing, while on those same days they would also get neurofeedback training. And of course, we measured their functions before and after. And so we looked at executed functions using like a task battery. But we also took functionality measures, which basically is like asking the participant for what do you actually want to improve, why are you here, did you achieve that goal, yes or no, something like that. And we also looked at general placebo and expectancy effects, which all looked surprisingly fine for whatever reason. And this is what we found. So this is only from the neurofeedback group. So this is the neurofeedback data we have, we've gotten out of this. So I will walk you through. So what you see here is basically the averaged trend of how this would go. So you start over here at the start baseline. This is the very first time you come in. And then you have like your 5 blocks of training on a Monday. And as you can see here, the people, they didn't really change much at all in the beginning. Then towards the end of their first session, they kind of got a little bit better. And then towards the end, this flattened off a bit. Now, 2 days later, on Wednesday, they came back. They kind of already knew what was expected of them. They also figured out what training worked best. And as you can see here, they were quite well able to upregulate their theta. And then towards the end, it went down again. Maybe they're tired. Maybe this was completely just like a random fluke. We don't know. And then towards the 3rd session, you see this becomes a bit more stable, so there is still an increase. And when you just look at this from, when you average each session into 1, you can see there is like an increase, like this is a medium effect size, almost significant. We had a small sample. We have no control group. Is this better than only doing neurofeedback? I can't even tell you at this point. the main focus we had was, does this work at all? And well, we got some data, which is good. However, how does that translate to the behavioral data? Not so much. So did the people actually improve on the tasks we were giving them? Nah, not really. So on the left you see, well, before, and here you see after, and yeah, well, that didn't work out so much. However, when we look at the self-report data, which of course, again, take this with a grain of salt, people reported that they were actually feeling as if they would be doing better on working memory, state shifting, monitoring, inhibition. So interestingly, like we compare this to like the people who also microdosed, right? So like the comparison here is all of them microdosed. However, the people who got neurofeedback and microdosing, they were doing even more better than the people who only microdosed. But then again, neurofeedback also has almost the same amount of expectancy bias as psychedelics have, so therefore this might be just like a synergistic effect on placebo. Who knows? Well, in terms of feasibility, we also asked the people, why are you here and what do you hope to get out of this? And did it actually work in the end? And we did also find that the people hoping to improve their cognition said, at least afterwards, they were doing a lot better. Same goes for presence, same goes for mood. So the people liked it. Like, we had no dropouts, the people were fine. We managed to do this type of research, so we kind of showed that this is feasible, that you can try this out. So yeah, overall, mixed bag, but I was pretty happy. It was the first attempt, considering that we had no money. I was still, I think we got a cool paper out of it. Right, quick discussion. So again, strength, semi-naturalistic study design, which is great because we could do like a psychedelic trial in which we had like an experimental group and like a control group with virtually no funding. So semi-naturalistic study design in the sense of that the people took the substance at home, they sourced the substance themselves, yet they came into the lab and we could measure them. And well, we also kind of had control over the substance they were taking because we were, of course, collaborating with the guys who were providing this. It's a bit unofficial, but yeah. So we also had a good idea of what substance they got, that the truffles were fresh, that they were all from the same batch, stuff like that. Well, and of course we showed feasibility, which is great. However, well, the same limitation cuts both ways. Semi-naturalistic study design, of course, means that we don't have as much control as we would have in a clinical trial. For feasibility reasons, that was not possible. Then we lack a placebo control group, and well, we have no way of telling whether or not this effect we see is actually related to neuroplasticity at all or something completely else. But ultimately, the reason why we did this to begin with was to apply for a grant. So we just wanted to see if this works for us, and then we wanted to show someone who might give us more money, hey, we can do this, so give us some money and we'll actually tell you what's going on. So yeah, with that, I'd like to round off with this little quote. I found the picture and then I was like, fuck, I need a quote. That's the way it actually went. So what we don't know is much more than what we know. This is definitely what it feels like to work in neuroplasticity research right now as a second-year PhD student. Yeah, so with that, thank you so much for your attention. Right, before we come to the questions, I have a bit of advertisement to do as well. So my study is this close to actually kicking off. So today I would like to officially open my recruiting process. So if you are over 55, or if you know anyone over 55 who would like to participate in research involving neuroplasticity in Fribourg, well, you can contact me or you can go on our website and you can find all the information there. Also, my professor asked me to tell a bit about that. We also take interns. Right now, we are in desperate need of someone who knows how to work with heart rate variability data. So yeah, if that is something for you, you can contact him, contact me, take the link. Cool. Okay, now I'm taking questions.
Interesting talk and congratulations on your publication. I have a question regarding modulating neuroplasticity overall. From my understanding, as soon as you modulate plasticity in a tissue, you have a potential risk of causing cancer. Do you know if there's any research regarding psychedelics?
So there's no direct research involving that, but we know for, I mean, we have a lot of data that psychedelics do not cause cancer. I mean, let's put it the other way around. We have no evidence that would suggest that psychedelics do cause cancer. So therefore, given the fact that LSD in particular is very popular for like several decades now, I don't think there's been any association made between LSD consumption and risk of cancer. So I think that in terms of neuroplasticity, it's a specific type of neuroplasticity. Not all neuroplastic changes are the same. We simply don't know enough right now at this point to pinpoint exactly in what part of the brain psychedelics induce neuroplastic changes. We don't even know if this is actually true in humans. We still kind of try to figure that out. So remains to be seen, but probably not.
Thank you.
You're welcome.
Hello, thank you very much for your talk. I have a question. Why didn't you use neurofeedback only as a control group?
So feasibility mostly. So originally, when I set up this whole study, all I wanted to do is get a couple of friends, come to the lab, let's see if we have the technology, if we can actually do this. And then my professor got all hyped about it and was like, let's do like a full-blown study. And we very soon realized we're kind of a bit out of depth because we had no people, no money, no nothing to actually do this. So we had to come up with a design that shows the potential maximum effect. Because that is usually how you apply for funding. And if you would already choose the comparison between neurofeedback versus neurofeedback and psilocybin, the effects are probably a lot smaller than if you would look at nothing versus neurofeedback and psilocybin. So this is like a strategic, almost like political reason. But yeah, of course, like the study that hopefully will kick off at one point in the future will be doing a full comparison. So all 4 different types of combinations, placebo, neurofeedback, placebo, psilocybin mushroom, neurofeedback, and like psilocybin mushroom, and like a sham neurofeedback, basically.
Thank you for your talk. So I'm just trying to make an hypothesis around this to merge potentially different works together. So you say that participants felt they got it better while it was not the case in reality. So could this be coherent to say that they felt their own metacognitive feelings of, I made it right, more amplified, but then in the end it was just a subjective effect because of the psychedelic?
So the main thing with these cognitive tasks is that, of course, your performance on the NBAC, for example, or like the task switch test, or like the stop signal task, do not necessarily reflect your innate understanding of what working memory is all about, or how you are able to use cognitive flexibility in order to do your day-to-day stuff. So I'm not saying that these are completely unrelated, but we do see that there is a bit of a disconnect between objective task performance and subjective measure of executive functions. So the point I try to make is also looking back at the population we were investigating. A lot of these guys were students, and we kind of always had the problem that we more or less started the whole sampling duration within their exam period. And these guys told us that actually the microdosing helped them to get through their exams. So for them, it felt like they were actually doing a lot better, maybe only because of the slightly stimulating properties. So they had to drink less coffee, they were more awake, they were a bit more mindful, stuff like that. So, but yeah, of course, the hypothesis you put up is as plausible as all the other hypotheses. We can't really say at this point.
Yeah, thank you. I had another question.
Okay.
Could you imagine one day being able to, like, I don't know, like, because I really don't understand too much neurofeedback, but it's super interesting as a tool, right? So could you think, like, normally, for example, what we can do, as I understood it, you can, for example, navigate a small space shift and keep it in the center of the screen. So, do you think we could be able to like correlate the neurofeedback effect with the subjective feeling? So in order to keep, for example, an object very clear in high definition on the screen, you would have to keep being focused and amplify a given feeling you have. For example, concentrating in your own sadness or happiness or your own eureka feeling, and while doing so you keep it working, you make it work, so you have a feedback loop that makes you amplify and basically being able to learn how to modulate and create that feeling within yourself?
So I can say 100% yes, based on the fact that they actually have already done that. So there's been this research group in Tel Aviv, which is basically they're more or less like the heroes of the whole neurofeedback scene, because what they were doing is they took people who were suffering from obsession compulsion disorder, in particular when you had like a thing with cleanliness. They would put you in a bathtub while at the same time giving you augmented reality goggles. And then they would simulate that the bathtub and the whole bathroom, the whole room in which you are in, is dirty. It's really, really dirty. And the way how they train neurofeedback is by basically your anxiety levels correlate with your levels with the level of cleanliness in the room. So if you manage to downregulate your anxiety, then the room becomes cleaner. And it's genius how they gamify this process, and how you do therapy in such a way. Like there's such a strong conditional response in that measure, because you actively reduce like a negative stimulus by reducing your anxiety, and like this learning effect is amazing. And so yeah, you can definitely do that. Also, when we look at AI technologies, when we look at big data stuff, so they looked at fingerprints using EEG and combining that with MRI data. So they looked at different cognitive constructs like anxiety or feelings of disgust. How would that translate to EEG data? Because usually this is like more amygdala regulated stuff, so you can't really pick that up using EEG, but they found a way to make this work too. And the more data they have, the more interesting the algorithms might become, and the more applicability this EEG-based neurofeedback might get to. So yeah, it's very exciting to do neurofeedback stuff. I kind of stopped doing it right now, but maybe I will again in the future. Yes.
We have time for one last question.
How easy do you think will it be to assume that the results you can see through the experiment in the lab is something that could also affect people in a more complex behavior? And how far could you tell that maybe the effects you see are somehow restrained to specific brain areas, rather than a dynamic interplay, for example, when it comes to behavior or symptoms that are shown through particular behavior that you actually want to change through therapy?
So if I understood you correctly, you want to know about the ecological validity. So what we measure in the lab, how does that translate to the outside? Well, I mean, we kind of try to do that by really asking the people, hey, how you doing? Are you getting better? Basically just kind of keep it more of a loose structure and give a bit more freedom in what they want to respond. And basically through that, we just generate a lot of data that is a pain in the ass to analyze. But ultimately, this gave us a lot of insight on why actually people come and what happened to them afterwards. So this is one potential way to at least have an idea what's actually going on. Because you were right, like the big problem is usually ecological validity when we have these type of sterile lab environments, specifically when you look at this tiny sub-component of a specific sub-executive function thing. What does it actually mean in terms of do the people get better? Yeah, that's a very good question. Yeah, ecological validity, very important.
Thank you again, Morten, for the talk.